Hey, Derek.
Hey, Diane.
So you and I are about the same age. Tell me, do you remember the Terry Fox Marathon of Hope?
Sure, I think everyone who was Canadian who was around back then has that image of Terry running down the highway on his prosthetic leg burned in their memory.
Yeah, I feel the same way. I remember it was super riveting, and also I remember how sad it was when finally he had to stop running and eventually he died. And another thing I remember about that was that what it felt like to me was that we were told that he was running to find a cure for cancer. Not a specific kind of cancer, but just a cure for cancer as a whole.
Yeah, that's true. I think for many years we always thought that cancer was just a single disease and that one day somebody would find the master cure.
Yeah, and we now understand that cancer is far more complicated than that. Not only are there many different kinds of cancer that are almost different diseases from one another, but cancer functions really differently in every individual person. So trying to treat cancer in you or in me or in anybody else is a completely different project.
Yeah, but I understand there's some really exciting work happening right here at McGill through the Goodman Cancer Institute and other places on campus. And I think you've got an interview today with someone who's leading that charge.
I do. I was lucky to get to sit down with the brand new director of the Goodman Cancer Institute, John Stagg. He's a really well-known cancer immunologist, and we had a great conversation about what cancer really is. Is it one disease? Is it many diseases? What some of the most promising areas of research and treatment are, and where we can all think that we can find hope for ourselves, even if it isn't exactly the kind of hope that the Terry Fox Marathon was about.
Well, that sounds like it could be a real shot in the arm. So let's cue the interview and hear what Professor Stagg has to say.
Welcome to the Mark My Words podcast. Today I have the pleasure of speaking to Dr. John Stagg. Dr. Stagg is a cancer immunologist, a professor at McGill's Faculty of Medicine and Health Sciences, and the newly appointed director of the Goodman Cancer Research Institute. In fact, John has barely had time to hang up his lab coat because he just started this week at McGill. He comes to us from the University of Montreal's Faculty of Pharmacy, and we are delighted to have him here at McGill. Welcome, Dr. Stagg.
Oh, thank you, Diane. Pleasure is mine.
So maybe I'll start by asking you a little bit about your background. Maybe you can tell me where you're from, what drew you into cancer research, and what brought you back to McGill.
Of course. So like many of us, I was personally affected by cancer. Growing up north in Chicoutimi, one of my aunts died of cancer before she turned 40. And she was actually my godmother, so I was really close to her. And it really affected me personally and the whole family. So growing in this really tightly knit family with someone affected by cancer, and when I was at a young age, really drew me into medicine, into cancer research more specifically. And at some point I thought of becoming a medical doctor, but what really was of interest to me was the discovery process, discovering new things that are unknown right now. So I ended up doing my PhD in experimental medicine here at McGill University. So I'm actually coming back to my alma mater. And so I graduated from McGill University with a PhD in 2005. And I was studying at that time an emerging field called cancer immunotherapy. And I was lucky to be mentored by an amazing clinical scientist called Dr. Jacques Galipeau. And Dr. Galipeau was a passionate doctor, really close to his patient and really close to his students. And he transmitted that passion to me. So after training with Dr. Galipeau, I moved to Australia to do my fellowship, so specializing in cancer immunotherapy. And I returned to Montreal in 2010, where I opened my lab at the CHUM Research Centre, part of University of Montreal.
Excellent. Well, first of all, always happy to hear of people who were really inspired by their experience as students at McGill. I've heard that from lots of people who've ended up back here, and obviously also wonderful to welcome you back. So I want to get more into this question of immunotherapy, what it is and what your own research is leading you towards there. But before we get to that, I thought maybe we could just take a step back and talk about kind of the state of the understanding of cancer right now. I was talking to a colleague and we were saying that when we were kids, people were always talking about finding a cure for cancer, like it was one disease and someday somebody would wake up and find a cure. Then we sort of entered a world where we began to understand that cancer was many different diseases diseases or disease mechanisms operating in many different ways. And now we seem to be at this moment where it almost feels like precision medicine is telling us cancer is different in every single person and we need personalized therapies. So I guess one way of asking the question is, how do we understand cancer right now? Is it one disease? Is it many diseases?
Yeah, that's an important point. So as you highlighted, cancer is not one disease. It's an umbrella of hundreds of different diseases, all of which share one common feature, like you said, that cells start dividing and invading surrounding tissue. But it's more complicated than just uncontrolled cell growth. Each cancer arises in a different tissue and has a different fingerprint, a genetic or a mutational fingerprint. So each person's cancer has this unique fingerprint, basically. So the two people with the same type of cancer, let's say two people with lung cancer, will have very different cancer, in fact, because of those different fingerprints. So that's why in modern era, we say that every cancer is unique to every patient. And personalized or precision medicine comes from this realization that instead of one-size-fits-all treatment, well, we need to tailor therapy to each patient.
So given that understanding now of what cancer is and how it operates differently in people, maybe that's a good entree to actually start talking about immunotherapy and your own research in the field. First of all, what is it? And maybe you can talk about what you do and also what you see as being some of the most promising avenues that immunotherapy is opening up for cancer patients.
Absolutely. So immunotherapy is an approach that uses the body's immune system to attack and destroy a cancer, basically. So for decades, we've known that the immune system can sometimes do this by itself. But unfortunately, the tumor also adapts to these attacks from the immune system and finds a way to either suppress or to be silent from the immune system. So the real breakthrough came about 2 decades ago with the discovery of so-called immune checkpoints. So immune checkpoints are really natural breaks that the immune system has to keep balance. But like I said, the tumor evolves to harness these breaks to evade the immune system. So cancer cells exploit these breaks. So drugs that now block these so-called immune checkpoints release the breaks and allow the immune cells to come in the tumor and kill the tumor cells. And for some patients, this has been nothing short but life-changing. I'm thinking of patients affected by metastatic melanoma or some patient with lung cancer, which now really, really, truly benefit from these groundbreaking discoveries. But unfortunately, for the majority of cancer patients, that's not the case still. Today, most patients still show resistance to immunotherapy. That means that their tumors still have ways to evade the immune system. So that's what my lab studies. My lab studies these new mechanisms of immune evasions that are still unknown to us.
I guess one question that raises for me is cancer evolving sort of constantly to adapt? That is to say, as we develop new immunotherapies, are cancers going to evolve to evade those too? Or is this a sort of adaptive mechanism that sort of took place in the past and now things will allow us to address it going forward?
That's an excellent question. I think that's one of the most important questions, actually, because we know that the tumor is an evolving entity, like you just said. The advantage of our immune system is that it can also adapt, right? So we have an adaptive immune system that allows us, for instance, to respond to infection that itself, infection agent also evolve over time, over different cycles of infection, and our system can also in parallel evolve. So the key is to find ways to induce this co-evolution or this co-adaptation of the immune system in parallel with the tumor evolution, if I may say. So up till now, like I said, we've identified some immune checkpoint or some breaks that the tumor exploit to block the immune system. But I'm confident we can find more of those breaks that can be targeted. But I'm also confident that we can identify, for instance, mechanisms to promote immune memory of our immune system towards these emerging cancers. I think one of the most exciting avenues at the moment is the development of cancer vaccines. So there's two types of vaccines, really. There are the therapeutic vaccines that the patient gets once they have the diagnosis. And there's the prophylactic vaccine that you could get if you are at risk of developing a certain type of cancer. The first cancer vaccine that comes to my mind is the protective vaccine, is the vaccine that protects you against HPV infections. And to come back to my godmother that died when I was young, she had cervical cancer due to HPV infection. And if she had lived in the 2000s, she would have been protected because she would have received that vaccine against HPV. And that would have protected her from developing cervical cancer. But that's just one type of cancer. What's emerging now is therapeutic vaccines, where we can identify the fingerprint of your tumor and develop in a relatively short time a personalized vaccine to you. And that would stimulate your immune system to recognize those fingerprints and destroy the cancer. So I think this is really, really groundbreaking research being done right now, including in Montreal. I mean, there's clinical trials going on here at McGill, at the CHUM, and other hospitals. So this is very exciting. Another field of exciting research for me is the development of cell therapy and cell engineering. So we've seen in immunotherapy really two major approaches over the years. One is targeting these immune checkpoints I was mentioning before, and that is done by injecting to the patient antibodies that block these immune checkpoints. The other big development or big breakthrough came from the development of cell therapies, so-called chimeric antigen receptor T cell therapy. These are very laborious therapies because they require that each patient go into a long process of having their cells taken from their blood, gene engineered, and reinfused into them. What we think the future holds is technologies are rapidly evolving, and what we think the field is moving towards is the possibility of engineering those cells within the patient without having the patient needed to have their cells extracted and cultured in the lab. So we're seeing now technologies where the medicine can be injected to the patient, and that medicine can, in the patient, engineer the immune cells to then recognize and kill the tumor cells. And these technologies that re-engineer the cells in the patient are at the moment developed by the use of RNA technologies. So that brings me to the other point, which I think is RNA technologies are here to stay, and beyond the vaccines that we all know, there's a lot more that can be exploited from these RNA technologies.
I think that's super interesting. On the one hand, I think thinking about sort of harnessing the body's own capacities, the adaptability of the immune system and other things is, a kind of, it feels exciting to think of exploiting mechanisms that already exist and just finding creative ways of sort of harnessing those to fight cancer. And it's also interesting to know that cancer is one of these fields where RNA technologies beyond infectious diseases, for example, or other things we might be thinking about, has so much potential. A lot of what you're talking about is the development of therapies, many of which are probably drugs, which kind of leads to a question, I think, that sort of asks about universities sitting at the intersection of very basic research and drug development. So your work has led to clinical trials with pharmaceutical companies, so you're really well aware of kind of how this works. So maybe you can talk about what some of the hurdles and opportunities are in translating the basic discoveries that you make in your lab or that others make in their labs out into the world so that they actually become drugs that we can all take.
Yes. So I've had the chance, the opportunity to be involved in a startup a couple of years ago. I was co-founder of a company that's called Surface Oncology, and it was a very exciting time to be in that space and to take my basic fundamental discovery and translating it into a drug development program. That was really, really exciting. But like you said, there's challenges to do this. So moving from a discovery in the lab to an actual treatment for patient is a long and complicated and expensive journey. We can make this very exciting findings. We discover new molecules in the lab. We discover new ways that tumor grow, or new ways to boost the immune system. But turning that discovery into an actual medicine requires years and years of testing, safety studies, clinical trials, regulatory approval, and investment. So one of the biggest hurdles in this journey is what we call the valley of death, which is the gap between academic discovery and commercial or drug development. Universities are fantastic at making discoveries, but they usually don't have the infrastructure or resources to carry them all the way to clinical trials. So that's why partnership with industry, with biotech companies, with venture capital firms, and also with philanthropy organization is essential. So they provide the expertise and resources to scale up discovery into something that can really be beneficial to the patient. So I would just add that another challenge is the mentality, the mindset. As scientists, we're trained to make discoveries and to publish our discoveries in the highest journals possible. The drug development process is based on business model, regulation, and clinical activity. And bridging those two worlds really requires people to speak the same language. And that's one challenge. Often I see that the scientists struggle to explain or value their discoveries to the drug developers, or even to sometimes they don't see how the process works. What are the different steps that would lead to phase 1 or phase 2 clinical testing? And at the same time, the drug developers sometimes look at scientists like weird beasts, and that they don't really know what drug development is. But if scientists would be better trained at drug development, I think that would be fantastic. So that's why I think we need to train the next generation of researchers so that they're proficient in this know-how of drug development.
I've heard other people mention both of those hurdles that you've described there. We've certainly been lucky to have some donors who have given us funding at McGill for something called the McGill Innovation Fund, which is meant to help cross that valley of death and help researchers bring their discoveries further along so that they might be more able to test them, pilot them, and see if they're ready for market. And the other is this question of training researchers who can speak the language of industry and be able to play that role a little bit more effectively. So maybe you could talk a little bit about what you think that training looks like and how we either are doing it or might be able to do that at the Goodman Institute to sort of produce people who can operate in that field really well.
Yes, absolutely. So like you said, it's important to train the new generation to be versatile and into these different spaces. So it used to be that you need an ace in your card deck, but now it's about having a full set of skills in your deck. So you need to train these new students and postdoc not only in different scientific fields, but also across the drug development process. And so I think it's important to integrate, for instance, the schools of management here at McGill with the School of Biomedical Science. So I think that's important to have integration between the two. And another way to achieve such training is to offer internships in industry, or to partner with industry to help them in their drug development. So many of the biotechs at the moment are struggling to raise investments, and so perhaps it's an opportunity for academic labs to partner up with them into their drug development venture, and that would also offer a chance for our trainees to get exposed to industry here in Quebec. And like you said, there's also a need, I think, to stimulate entrepreneurship and startup creations within McGill. That, I think, will require a commitment from the organization, and with the support of philanthropy, I think we can achieve it. So I think it's important to rethink the way that we value our discoveries. And I think one of the new ways that we can value our discoveries is by getting the donors to participate in this entrepreneurship venture, I think.
Yeah, and I think we have many people in the McGill orbit who themselves come from an entrepreneurial background, and so are sort of keen to help the university, move in a direction that allows us to really, as you say, value our discoveries differently. So maybe I'll come back to your new role at McGill, or coming back to McGill, a homecoming of a certain kind. Maybe you can talk a little bit about what is unique about the Goodman Cancer Research Institute. What role does this entity play in the sort of cancer research ecosystem in Quebec that you've obviously been a part of for most of your career? And what, in your view, is some of the most exciting work that's going on there?
Mm-hmm. So yes, Diane, I'm super excited to be the new director of the Goodman Cancer Institute. The Goodman Cancer Institute really represents a powerhouse of cancer research at McGill University. So over 250 trainees, 25 members physically present within the building, with 15 other associate members in other locations, all dedicated to translate basic discovery into new treatment for cancer patients. And that's really the vision of the Goodman Cancer Institute, to make an impact to the patients. And so my hope as director is to build on that foundation and push it even further. I want the GCI to be a hub of innovation, a place where ideas are nurtured, where discoveries can move from the lab to the clinic. And I see the GCI as one of the main drivers in Canada that can connect those efforts. And honestly, the GCI has all the ingredients to achieve this. Outstanding scientists, outstanding trainees, strong infrastructure, strong support within the School of Biomedical Science, and a history of excellence. 2025 actually represents an important year for the GCI as we mark the 60th anniversary of a very important discovery in cancer, which is the discovery of a diagnostic marker called the carcinoembryonic antigen that was made by Dr. Phil Gould 60 years ago. And Dr. Phil Gould was the director at the time of the McGill Cancer Center, which is now called the Goodman Cancer Institute. So my job now is to grow our international collaborations and to make sure that the discoveries that we make are translated into real benefits for cancer patients.
That is, exciting to hear. It's always, I think sometimes we can sort of forget about this history that a lot of these entities at McGill have. And it's kind of amazing to think that exactly 60 years ago, some very important discovery was made that's still informing cancer research. And then you can sort of pick up that mantle today. So I think I'll just ask you one last question. Cancer research, I imagine, is a long and often challenging journey with slow progress and things that seem promising and then don't come to fruition. Yes. And of course, there's a lot of suffering whenever you're dealing with people who are living with cancer. So what keeps you motivated, and what makes you feel like within your tenure at the GCI, we're actually going to see some really important improvements to how people can get better and perhaps even be cured of different kinds of cancer?
Yes. Research is a marathon. So experiments fail, trials take years, and progress can sometimes seem painfully slow. Yeah. But what keeps me motivated is the people behind the science and the patient that need the new treatments, really. And also, as motivating is to see the patients now that benefit treatment that did not exist 15, 10, 5 years ago. To see this patient living well, and and imagining my godmother back when I was a kid, who might have lived a better, longer, healthier life if she had been alive now. So that keeps me motivated. And to be honest, the hype of discovery, there's nothing like running an experiment or having a student coming to you with a new result and thinking that you're the only person in the world that knows that result. You know, that's a feeling that is hard to beat. It brings you endorphin. It's like a drug, and then you want to do it again. So that's really what keeps me going. So we're really in a golden era at the moment of technologies, and that opens doors that were unimaginable decades, just even a decade ago. So combine that with the passion of young scientists and their energy, I'm convinced that we will achieve breakthroughs that today that can only be dreamed about.
Well, I'm super excited that you're here. I'm sure I'm not the only one. And I can't wait to maybe redo this interview in a couple of years and have you tell me about all of those, exciting first discovery moments that you've had at the GCI with students and with other scientists. So thank you again, Dr. Stagg, for sitting down with me today. Mark My Words is a production of McGill University Advancement in partnership with Communications and Institutional Relations. Special thanks to our producer, Frank Roop, and to my co-host, Derek Kassoff, who will be back with you next week. New episodes of Mark My Words drop every Wednesday, and you can find us on Apple, on Spotify, or wherever you get your favorite podcasts. And you can also watch us on YouTube. And if you subscribe, you'll know that you'll never miss a new episode. Thanks very much, and we'll see you next week.